Hey everybody, welcome to week 8 of class. This week we'll be going over chapter 4, which covers cellular anatomy. And then next week we'll be covering, or rather 2 weeks from now, we'll be covering human anatomy. And I'm during these next 2 weeks, or 3 weeks rather, it's 2 lectures. I'm really hoping that you guys will see a similarity between the anatomy or organelles that you will find in a cell, and then the organs that you will find in inside a human, just so you can see that there is both a similarity, and hopefully it can help you guys remember the functions of each of the organelles and organs. So we have several hundred different types of cells in our body, and over 1 billion copies of each. We have nerve cells to conduct information throughout our body, gland cells to secrete hormones, bone cells that provide support for our body, and you guys can see all of these through a light microscope. And these light microscope can see frog eggs, plant and animal cells, bacteria, and even some of the organelles that we'll talk about later in this lecture. However, light microscopes cannot highlight everything within a cell. So during the 1930s, the electron microscope was invented. And electron microscopes are able to highlight all of the details that are inside of the cell, including individual molecules, just short of an atom. And this is really important for scientists to determine how different molecules are used within a cell. So cell needs to be able to make nutrients and excrete waste, just like we do. If our bodies were made of large cells, we would die. This is because the surface area of the cells wouldn't be able to take in enough nutrients to help maintain our body. So surface area is key. We want all of our cells to be really small, which would greatly increase the surface area of the cell membranes within our body, so they can take in more nutrients. Certain other adaptations besides just small cells that helps increase surface area can be found within our intestines. So our intestines kind of look, I suppose you can, they kind of look like this. I'm not sure of a good comparison, but on these, I guess maybe arms is what you could call it, are tiny other tiny arms, and these are called microvilli. And microvilli are also really good at increasing the surface area of our intestines, so that more of the nutrients that we get from the food are able to be absorbed within our body, so that we can make most of the food that we eat. So all cells have a plasma membrane surrounding them. It plays an important role because it regulates what goes in and out of the cell. And the cellular membrane is made up of phospholipids. And the phospholipid molecules look a little bit like tadpoles, with the heads being hydrophilic or water-loving, and the tails being hydrophobic or water-fearing. The side of the cell, the inside of the cell is a watery substance, so it's going to be hydrophilic. So the hydrophilic heads face inward toward the cytosol, or pretty much toward all of the insides of the cell. The heads also face outward of the cell because the cell is also going to be surrounded by a watery substance. The inside of the cell, though, is hydrophobic, which again just means water-fearing. So it's going to repel anything that's hydrophilic, or that has a negative or a positive charge. So you guys know how fat and water can't mix. This is because water is hydrophilic and fat's going to be hydrophobic. So you guys can more or less think of the inside and the outside of the cell as water, and the inside of the cell as fat. So they cannot mix with each other, and in the same way oil and water cannot mix if you pour them into the same glass. And because of this, because of the different because of the different chemistry of the tail and the head of the phospholipids, this allows the cellular membrane to be picky on what kind of molecules go through the membrane, which is really important in helping maintain homeostasis within the cell. And because certain molecules cannot go inside or outside of the cell, transport proteins are required. So here is the structure of the plasma membrane. Besides the transport proteins, which are these big purple structures right here, you will also find glycoproteins within the cell, which is going to be this thing. And these are responsible for cell-to-cell communication and will provide structural support for the cell membrane. Glycoproteins are also important for reproduction because they allow for the binding of the sperm cell to the surface of the egg. So also within this picture, you can see the phospholipids. The red heads are going to be the hydrophilic heads, facing both on the outside of the cell, out here, and on the inside of the cell, in here. And then these yellow tail-looking things are going to be the hydrophobic tails. Notice how there's two different layers of phospholipids, so we call a cell membrane a phospholipid bilayer. So again, those hydrophilic heads are going to be facing in and out, and those hydrophobic tails are going to be facing toward each other. So molecules that are not able to travel through the membrane will use these channel proteins. They will flow down their concentration gradient, which just means that there are going to be more molecules which just means if there are more molecules on the inside of the cell, then the molecules will flow into the cell. And this is the same, this is also true if there were more molecules on the inside of the cell than on the outside of the cell. Pretty much, the molecules will flow in and out until it reaches a balance in between the inner and the outer part of the cell. So essentially, there's going to be the same amount of, let's say, water on the inside and the outside of the cell. Another example, so proteins that help water flow in and out of the cell are called aquaporins. And one organ that uses these are kidneys. And your kidneys are reliant upon aquaporins because it helps regulate the amount of water that you lose in urine, or the amount of water that stays within your body. So aquaporins are really helpful in keeping your body hydrated and help concentrate your urine. Molecules that use transport proteins flow against their concentration gradient. It doesn't matter how much of the molecule is on the inside or the outside of the cell will bring it in and out as needed. Because the molecules flow against their concentration gradient, it requires energy to transport them, since it's not the natural tendency of the molecules to flow against their concentration gradient. For example, a transport protein helps sodium and potassium ions travel across nerve cell membranes. And this is really important in nerve conduction. And without this particular protein, your nerves would not be able to allow your mouth to move, your eyelids to close, or even your hands, arms, legs, and your toes to move. So again, really, really important transport protein. Another type of protein that can be found within the cell membrane are called receptor proteins. And these allow signal molecules to bind, which causes a cellular response. An example of this is when the hormone insulin will bind to a receptor protein on the liver cells. And this will cause the liver cells to take up glucose from your blood. And this is really important for any time your blood sugar is low, because then the liver cells will release glucose, raising up your blood sugar so that your blood sugar doesn't drop too low. And then we have enzymatic proteins, which directly participate in metabolic reactions. And without enzymatic proteins, the cell will never be able to perform important functions like degradation and synthetic reactions that are important to its function. Another type of protein we have in our cells are cell recognition proteins. And this enables our body to distinguish between our own cells and cells of other organisms. You can especially see this within cells of our immune system, like white blood cells and red blood cells. These are all of our different cells in our body will have certain will have recognition proteins that pretty much tells our body that our cells are our cells, so that our white blood cells won't go around attacking our various organs. Anytime a bacteria or a virus enters into the cell, then the white blood cells recognize that it's not our own, since it won't have our own personal recognition proteins. And so then the white blood cells would be able to attack the bacteria, and then make antibodies to it so that you don't get a future infection. And then we have junction proteins. And junction proteins will form junctions between cells and allow for cell-to-cell adhesion and communication. An example of this is the adhesion junctions in your bladder cells, which allows the bladder to swell with urine without breaking apart. And there are three types of junction proteins. We have adhesion junctions, found in the picture on the left-hand side. We have tight junctions, found in the center picture. And then we have gap junctions, which are found in the right-hand picture. So with adhesion junctions, plaques are going to be connected to the cytoskeleton of the cell for support, and then are connected together with intracellular filaments. So here we have our plaque on the inside of each cell, and then we have our intracellular filaments here, which connect the two cells together. Adhesion junctions are going to be important within organs that need to stretch, like the bladder, the stomach, and the heart. With tight junctions, the plasma membrane proteins connect together and form something like a zipper. Tight junctions are important for organs that serve as barriers. For example, urine stays within kidney tubules because the cells of the tubulars will be joined together by tight junctions. And this is really good because then urine won't leak into the rest of your body. And then gap junctions form when two identical cells join together by the lining up of the channels in the plasma membrane. As you can see, notice how these channels here have a column in it so that molecules can flow in between the two cells. These, though, still do provide some support for the sheet of cells. So this is important in heart muscles because the passage of small molecules will tell the surrounding heart cells to contract so that the entire heart pumps at once. So scientists have come up with the cell theory, which states that all organisms are composed of cells, and all cells come only from pre-existing cells. All cells have a plasma membrane to regulate what goes in and out of the cell. The cytoplasm, which is the inside of the cell, pretty much all the liquid and all the organelles, are where the chemical reactions occur. And then genetic material is needed for growth and reproduction, which of course is DNA. There are going to be two main types of cells: prokaryotic cells, which lack the internal organelles, more specifically like the nucleus, and then we have eukaryotic cells, which do have all the organelles, the more prominent one being the nucleus. So prokaryotic organisms will be found in the domain of bacteria and archaea. They are usually they are generally smaller and simpler in structure than eukaryotic cells, which is pretty good for them. It allows them to reproduce very quickly and effectively. And they are, obviously, because of them being able to reproduce so quickly, are going to be a successful group of organisms. And another reason, prokaryotes can be found in air, water, dirt, or in and on organisms. And bacteria is very important in decomposing dead organisms and returning those nutrients to the ground. So prokaryotic structure, bacteria being one of the more well-known prokaryotes, is going to have a cytoplasm, just like eukaryotic cells, surrounded by a plasma membrane. And then a lot of bacteria are going to have a cell wall, which just helps reinforce the plasma membrane, and also helps bacteria from anything that might try to destroy it. It is going to have a capsule, which is a protective layer. And the plasma membrane is going to be the same as eukaryotic cells, so that double layer of phospholipids. So it's going to still have DNA, except its DNA is going to be circular, coiled chromosome, which is going to be located in the nucleoid, which is a particular region of the cell and not necessarily within an organelle. But notice how the name is pretty similar to a nucleus. It's still going to have ribosomes, which are also found in eukaryotic cells, and ribosomes are going to be what creates the proteins that are needed for the cell from the DNA. And then it will also have appendages. Not all bacteria will have this, but some bacteria might have flagella, which are tail-like structures that help the bacteria move about, or it may even have cilia, which are more hair-like structures found all over the plasma membrane that will help the cell move, also move. So here you can see the basic structure of bacteria. You're going to have the cytoplasm here. All the little red dots are going to be the organelle, or the ribosomes. The ball of purple yarn in the center is going to be the DNA, located in that nucleoid region. You're going to have the plasma membrane that is touching the cytoplasm. Next is going to be the cell wall, which is helping reinforce that plasma membrane. And on the very outer layer, you're going to have that capsule. And then, so again, not all bacteria, but some bacteria will have flagella, which are going to be these tail-like structures here that are going to generally spin in circles to help the bacteria move from one area to another. So now we move on to eukaryotic cells. And I'm sure eukaryotic cells are going to be more familiar to you guys because pretty much any animal or plant that you can think of are going to be made up of eukaryotic cells. Eukaryotic cells can also be found in protists, which protists are similar to bacteria in that they're single-celled organisms. They're just going to be much more complicated. Eukaryotic cells are going to contain membrane-bound organelles, more prominently the nucleus. Because they have all these organelles, they're going to be much larger than prokaryotic cells. And all of the chemical reactions that are occurring within the eukaryotic cell are going to be compartmentalized within the various organelles, just like the chemical reactions within our body are also separated from each other within our own organs. And there are going to be 4 categories of organelles. There's going to be nucleus and the ribosomes. There's going to be the endomembrane system. There's going to be energy-producing organelles, and then there's going to be the cytoskeleton. So here we have the general anatomy of a eukaryotic cell. So this particular cell is an animal cell, and it is not a plant cell. that's because it doesn't contain a cell wall and two other organelles. So here we have the mitochondria, which is responsible for making ATP, which is a source of cellular energy. Here you're going to have a lysosome, which is responsible for getting rid of anything the cell no longer needs. Here we have the nucleoli, which is responsible for making RNA. Here we have the nucleus, which of course is going to house our DNA, which is of course important for creating any proteins that we may need or also needed for reproduction. This here is going to be the rough endoplasmic reticulum, and this here is going to be the smooth endoplasmic reticulum. I may abbreviate these to the rough ER or the smooth ER. And then this here we have the Golgi apparatus. And then of course on the outside we're going to have the plasma membrane. These yellow lines here are going to be our cytoskeleton. And then just like within the prokaryotic cell, these little red dots here are going to be RNA. So here is a plant cell. So notice how the plant cell will contain these cell walls, which just help reinforce the plant cell. That's why bark, the stems from leaves, or even the base of a plant is going to be a bit harder to break, or is going to be a bit more rough than, say, the skin of your dog or your cat, because it contains that cell wall. But it's going to contain pretty much the same organelles as the animal cell. It's going to have the nucleus, the rough ER, the smooth ER, the Golgi apparatus, these lysosomes. The ribosome is going to contain the cytoskeleton and the mitochondria, but it's also going to contain two different organelles that the animal cell does not have. And this includes the central vacuole here, and then it also includes the chloroplast here. Notice how the plant cell has, three different things that the animal cells do not. It's going to contain two organelles, and then just that extra outer layer. And to help you remember them, they all start with C. So cell wall, central vacuole, and then the chloroplast. So first we'll talk about the nucleus and the ribosomes. And I want you guys to think of the nucleus as the brain of the cell. In the same way that our brain tells us what to do, the nucleus more or less tells the cell what to do. And it does this by telling the cell which proteins to make, which is really important to help the cell function. And of course, will contain all of the information it needs in order to reproduce. The nucleus stores genetic information in the form of DNA. When the cell is not reproducing, that DNA is uncondensed, which is called chromatin, and then it will also contain proteins and some RNA. And right before cellular division, that RNA is going to compact into chromosomes, which we talked about the other week. DNA is organized into genes, which code for various proteins that our cells would need. And info is going to be passed from the DNA to ribosomes through three different kinds of RNA. And there's going to be messenger RNA, there's going to be ribosomal RNA, and then transport RNA. But we'll get to that in a future lecture. There's going to be the nucleolus, which was that dark purple dot that I pointed out in the previous two pictures. And this is the part where ribosomal RNA is going to be made. And if you guys remember those red dots in both the animal and plant cell pictures, those red dots is the ribosomal RNA. And then just like the cell itself, the nucleus is also going to contain a double membrane. And this is called the nuclear envelope. And just like with the cell, the nuclear envelope is going to allow certain molecules to come in and out of the nucleus. So here you can see the nucleus. So here we have our nucleoli, which is just going to create that ribosomal RNA. This little blue part here is that uncondensed DNA, also called chromatin. Just like with the cell, the nucleus is also going to contain that double membrane, allowing certain molecules to come in and out of the cell through these pores here. And then, so connected to the nucleus is the the rough ER, the rough endoplasmic reticulum. And this is going to be where certain molecules are made. Notice these red dots here. It's going to it looks, I'm sure, very familiar to the red dots out in the cytoplasm. So these red dots here are also is also RNA. So ribosomes carry out protein synthesis in the cytoplasm. So pretty much the formation of the proteins that are coded for in the DNA. As we discussed earlier, these can be found in both prokaryotes and eukaryotic cells. These are going to be composed of two subunits, one large and one small subunit. And ribosomes are going to be a mix of both proteins and ribosomal RNA. The ribosomes are going to receive the messenger RNA, where they are going to start making the protein. And in eukaryotes, they are going to be ribosomes free-floating within the cytoplasm, and then also going to be attached to the rough endoplasmic reticulum. So here you can see the basic process of protein synthesis. So here we have our DNA, and this other long blue string is going to be our messenger RNA. And our messenger RNA is pretty much going to contain a copy of the gene from the DNA that the cell wants, the gene that codes for the protein that the cell wants to make. The messenger RNA is going to connect to our transport RNA here. And then our transport RNA is going to connect to our ribosomal RNA. And the ribosomal RNA, or the transport protein, can connect either to a ribosomal RNA, either within the cytoplasm or on the rough ER here. And then the transport RNA is going to continue making our protein until it's officially finished. And when that protein is finished, it's going to disconnect from the transport protein and go wherever it needs to go. And the transport protein is going to disconnect from the ribosomal RNA and then go off to be used again. And then we move on to the endomembrane system. And the endomembrane system will consist of the nuclear envelope, the membranes of the endoplasmic reticulum, the Golgi apparatus, and the numerous vesicles. And this also helps compartmentalize the cell, making sure the chemical reactions only occur within certain areas of the cell. And so the numerous vesicles also include transport vesicles, which will carry molecules from one part of the system to another. So the proteins that the transport protein and the transport RNA and the rRNA just created is going to be within one of these transport vesicles to go wherever it needs to go. So the endoplasmic reticulum is a complicated system of membranous channels and saccules. And it is a continuous structure that connects with the nuclear envelope. So as I mentioned earlier, there are two parts to the endoplasmic reticulum. There's going to be the rough ER and the smooth ER. The rough ER is called so because of the ribosomes on the outside of the rough ER, giving it just that studded look. And this is where proteins are going to be made on the rRNA. And if those proteins need any type of modifications, then it will go into the rough ER where those modifications will be made. And then it will be put into one of those transport vesicles and sent off to the Golgi apparatus. The smooth ER is called so since it doesn't have the ribosomal RNA, and so it has a more smoother look. And it is, however, still going to be continuous with the rough ER. Instead of proteins, it is going to make lipids, like polylipids and steroids. And it will also, once these lipids and steroids are done, they will also be put into vesicles and sent off to the Golgi apparatus. And the function of the smooth ER will just depend on the particular cell it's in. For example, it will produce testosterone within the male reproductive system, and it will also detoxify your blood from certain drugs or alcohol that may be ingested. So here you can see both the rough ER and the smooth ER. Again, notice how it is one continuous system. Of course, the rough ER is called so because of the little red dots there, the ribosomes. Again, which is where the proteins are created. And if those proteins need any modifications, they will enter inside the rough ER within that yellow space there where those modifications will be done. And then the smooth ER doesn't have any of those ribosomes in it, and the phospholipids and the steroids will also be created inside of the smooth ER. And then the next part of the endomembrane system is the Golgi apparatus. And the Golgi apparatus is a stack of flattened vacuoles. To me, they kind of look like a stack of pancakes or a Spotify symbol. And this is more or less a distribution station of all the different molecules that the cell creates. They will receive the proteins and the lipids from the rough and the smooth ER. And if these proteins or lipids can still need further modifications, then it will happen within the Golgi apparatus. And once all the proteins and the lipids are finished, they will be put into vesicles again and sent off wherever they need to go. Some of the destination of where these molecules may go are the lysosomes, which are where these molecules may be destroyed. And this would only happen if the cell does not need any of the proteins or the lipids. And then I already mentioned it. Another part of the endomembrane system is the lysosomes, which are the lysosomes are vesicles that contain digestive enzymes, which again are going to be really important for getting rid of anything that the cell no longer needs, or anything that is just destroyed within the cell that will then the lysosome is also responsible for getting rid of any damaged products or particles within the cell. This is just to keep the cell healthy. You can kind of think of the lysosome as the immune system of the cell. Again, just making sure that it is getting rid of anything it doesn't need, getting rid of anything that is destroyed that could potentially harm it, and just making sure that the cell is healthy and clean. If a lysosome is not properly functioning, then a person could have something called Tay-Sachs syndrome. And Tay-Sachs syndrome is when the cell is not able to get rid of certain particles, and so these particles build up in the cell, and the cell eventually isn't able to function anymore. And this eventually leads to the person not being able to move anymore, and unfortunately they end up passing away. So here's a little bit more of a detailed description of Tay-Sachs disease. And this is when the lysosome is missing a certain enzyme that will destroy lipid molecules. And as a result, the cell becomes full of this lipid and stops functioning. And this can, besides just causing decreased muscle tone, causing the person not to be able to move anymore, it can also cause deafness, blindness, increased startle response, seizures, and delayed mental and social development. So here you have the entire endomembrane system. The rough ER accepts completed proteins from the ribosomes. The rough ER may then modify the protein, but then will send it off to the Golgi apparatus in a vesicle. The smooth ER makes lipids, like testosterone or phospholipids, and will send these off also to the Golgi apparatus within vesicles. The Golgi apparatus will accept these vesicles and will finish off the protein or lipid if needed, and then we'll send these vesicles off to their particular destination. Again, you can more or less think of the Golgi apparatus as the distribution part of the cell. The next organelle we'll talk about are vacuoles. And vacuoles are primarily going to be found within plants and protists, although they can be found in some animal species. There are many different types of vacuoles, but the main functions are going to be storage, digestion, and water expulsion. Just depending on the just depending on the organism that the vacuole is found in. In plants, the central vacuole will contain sugar, water, salt, and pigments like chlorophyll, which will give some flowers its red, blue, and purple color of the flower. Vacuoles in some animals will store fat like triglycerides, and these particular cells are called adipocytes. So here you can see vacuoles within two different types of cells. So within a plant cell, the central vacuole is going to be pretty obvious, as it's just going to be a large clear circle found within the cell. And then within a protist, the vacuoles are going to be a little smaller, but they should still be pretty distinctive. And then we'll move on to energy-related organelles. And one of those energy-related organelles that is not found within animal cells are chloroplasts. And chloroplasts will use solar energy to synthesize carbohydrates through the process of photosynthesis. And you can find, again, chloroplasts within plants and algae found in water. And chloroplasts will have a three-membrane system. On the outside is going to be a two-membrane system, which encloses the stroma. And the stroma is pretty much just like the cytoplasm, which is just all of the, which is going to be all the innards of the chloroplast. And then the thylakoids are going to be on the inside of the chloroplast, and this is what makes up this is what that third membrane is named. And the thylakoid membrane are going to contain pigments that will capture the solar energy needed for photosynthesis. So here you can see the basic structure of the chloroplast. So here you have that double membrane on the outside of the chloroplast. And then on the inside you have the thylakoids. And the thylakoids, it kind of looks like the Golgi apparatus in that it looks like a stack of green pancakes. The green color is given to it by the chlorophyll, and the chlorophyll helps capture the solar energy that is needed for photosynthesis. And then on the inside of the thylakoid is called the thylakoid space, while the inside of the chloroplast is going to be called the stroma. So another energy-related organelle is the mitochondria, and this is going to be found in both plants and animals. So mitochondria is going to be able to do a process called cellular respiration that is pretty much the opposite of photosynthesis done in chloroplasts. And so chloroplasts will make energy, while mitochondria will break it down so that the cell is able to use the energy within that original carbohydrate so that it can undergo its various functions. But we'll get into photosynthesis and cellular respiration in a future lecture. Just like the chloroplast, this is also going to have a double membrane on the outside of it. The inner membrane encloses the matrix, which you can think of as the cytoplasm of the mitochondria. And within the matrix is going to be a mixture of enzymes assisting in carbohydrate breakdown. And these reactions allow for the ATP synthesis. And ATP is going to be that cellular energy the mitochondria is going to be able to make through the carbohydrates through cellular respiration. So here you can see the basic structure of mitochondria. On the mitochondria, cellular inside and outside membrane are going to be this brown structure here for the outer membrane. And then the inner membrane is going to be this yellow structure here. On the inside of the membrane, of the mitochondria is going to be the matrix. And cellular respiration will happen in between the inner part of the double membrane and within the matrix. So the molecules are pretty much going to flow in and out like this, which is going to create a lot of ATP for the cell to use. And then we'll move on to the cytoskeleton. And you guys could think of the cytoskeleton as the muscles and the bones found within humans, as they create a lot of structural support and even allows for some movement within the cell. The cytoskeleton is a network of interconnected protein filaments and tubules. And these will extend from the nucleus to the plasma membrane. These are only going to be found within eukaryotes, and besides allowing for extra support and movement within the cell, they are also going to help maintain cellular shape. And motor proteins called myosin, kinesin, and dynein are going to allow the cytoskeleton to help transport certain things like vesicles around the cell. So motor proteins are going to include myosin, and myosin interacts with actin filaments and is going to be really important in muscle contraction. And these cells move in amoeboid type fashion. Amoeboid type fashion, if you guys can think of an amoeba, looks like if you ever throw a paintball at a wall and it has a splatter look with these different arms here. That's the shape of an amoeba. And an amoeba moves by creating these something called pseudopods, which is going to be one of the arms of that paintball splatter, and it's going to use that pseudopod to move it forward. So these myosin filaments proteins are going to do the same thing. Kinesin and dyosin, on the other hand, will move along the microtubules, and they help move the organelles around. And these are also really important in transporting vesicles to and from the Golgi apparatus. So here you'll be able to see how the kinesin transport protein moves vesicles around the cell. So we have the kinesin protein here, and it's going to contain this receptor. And the receptor is where the vesicle is going to attach to the motor protein. And because the motor protein is going to be moving these vesicles around the cell, is going to require ATP. ATP is a form of cellular energy, and ATP is what is produced during cellular respiration. Here we have the cytoskeleton made of those various filaments, and then the kinesin motor protein is more or less going to walk along the cytoskeleton, getting that vesicle to wherever it needs to go. And then we also have microtubules. Tubules. And microtubules are small, hollow cylinders, and they are going to be assembled and controlled by the centrosome. These also help maintain cellular shape and help as a track for organelles and other materials to move along. Microtubules make up the spindle fibers that are really important within mitosis and meiosis, just helping move those chromosomes from the center of the cell to the edges of the cell. So here you can see the basic structure of a microtubule. It is going to be two different strings of proteins connected together. And within it's going to be a hollow, more or less a hollow column of proteins. And the microtubules are going to be created by the centrosome, which are going to be very important within cellular replication, the movement of vesicles, and also for structural support for the cell. And then we have actin filaments. And actin filaments are kind of like microtubules in that they're also going to be made of strings of proteins. But these actin filaments are really important in structural support of the cell. They're going to be located just underneath the cell membrane. And these are also going to be really important within muscle contraction. So like I was saying a little earlier, microtubules are going to be created by centrosomes, and the microtubules are going to make up the centrioles found in those centrosomes. So here you can see the basic structure of a centriole, and here are the microtubules. They're going to be found in triplets located all over the centriole here. And centrioles are going to be found only within animal cells, and again are really important within cellular replication, more specifically DNA replication. Some of the motor appendages that we talked about earlier are cilia and flagella. These will only be found within prokaryotic cells, and these are really important for helping the particular organism move around the cell. Cilia are hair-like structures and move in a wave-like fashion and will be found all over the cellular membrane, while the flagella are more tail-like and are going to generally, you're only going to find 1, 2, maybe 3 flagella on a particular organism. And there are only going to be a couple of cells that will have cilia and flagella within humans. Ciliated cells can be found within our respiratory system, which will help sweep debris trapped within the mucus back up into the throat, which will help keep our lungs clean. And then the only cell you'll find with a flagella is going to be sperm, which is really important, of course, getting up within the female reproductive tract. And sperm are, of course, going to try to fertilize eggs. And eggs are going to be moved from the ovary to the uterus by ciliated cells within the fallopian tubes. Cilia and flagella are also made up of microtubules. In the flagellum, there's going to be a central pair of microtubules. Surrounding these are a pair of microtubules that are going to be connected by the motor protein dynein. This allows them to move around. Cilia move in a whip-like fashion, while flagella will move in a corkscrew-like fashion. Alright guys, thanks for joining me for this lecture. Hopefully it was pretty straightforward, and hopefully it'll be easy to understand. Should be just a lot of memorization. I hope you guys have a good fall break, and as always, let me know if you have any questions. Take care.